friends, welcome to the ITLS Academy. Today we are going to discuss the topic bioinstruments, the instruments which are used in our lab. The first instrument is microcentrifuge. As its name suggested that it is used for the separation. The microcentrifuge is a laboratory centrifuge. There are two types of centrifuge are present in our lab. One is centrifuge and other is microcentrifuge. Microcentrifuge is basically it is used for the separation of DNA, RNA, protein. DNA, RNA and protein. This is a laboratory centrifuge and it is used driven by a motor which is spin the liquid sample at very high speed. Due to this high spin, this liquid are separated the component, and it is due to the increasing the effective gravitational force will more rapidly and completely. It is called cause precipitation. There is a pallet. Suppose this is your append drop. In microcentrifuge we use append drop, and the liquid are present into it. When these append drop is put into a microcentrifuge and it move at very high speed, it is separated into a supernet and a pallet. The upper part is called a supernet and the lower part is called a pallet, and these pallet are maybe DNA, RNA and protein. Sometimes we use pallet as our further experiment, and sometimes when we use supernet as our further experiment. The rate of centrifugation is specified by the acceleration applied to the sample. The rate of centrifugation is directly proportional to the applied acceleration. If the speed is very high, then the separation rate will also high, and it is typically measured in a RPM, that is revolutionary per minute, or relative centrifuge force. This is all about the microcentrifuge. The second instrument is analytical balance. Analytical balance is simply it is used for the weighing the sample's weight. This is your digital analytical balance. We use analytical balance because it gives high precision or accurate result. In analytical centrifuge, the glass walls are present which enclose the sample inside the chamber, and this wall not allow the air to enter into the chamber. That's why it gives the very accurate result, and it is used to measure the 0.1 milligram. Even the 0.1 milligram weight is measured by analytical balance. See, analytical balance is used to measure a mass to a very high degree of precision and accuracy. The measuring pan of high precision, even it will measure 0.1 milligram or better. Analytical balance is inside a transparent enclosure with doors, so that it does not collect any air current in the room that do not affect the balance operation. If the air enter into the room, they will alter the result, but in that case there is no chance to enter the air and it will not alter the result. How we use analytical balance? See, when we switch on the button here, the value is shown 0. Now we put our aluminum foil or maybe sometime battery plates and tear it weight to 0. There is a button tear which convert the aluminum foil weight or battery plate weight into a 0. Now we put the sample weight into it, and after putting the sample weight, we close the door and wait for the equilibrium create. Suppose we have to weigh 24.2 gram of wheat flour. Now what we have to do? When we put the wheat flour into a aluminum foil, then we wait for to we wait for so that these weight become equilibrium. This is the use of an analytical balance. Now second is your pH meter. pH meter is used for the measurement of the pH. In our school days, what we have to do, we have to measure the pH with the help of pH paper. Now pH paper is not used, we use pH meter. Okay, so pH meter have a display button, have a temperature. See, suppose this is your pH meter, this is the display button in which the value are shown, and after that there is a temperature, then after that there is a function button, then there is a slope, then after that there is a button which is mentioned at instant by. Now what we have to do, and the last part is your electrode. This is your electrode. These electrode is always dipped into a KCl solution. That's why, because if the electrode is not dipped into a KCl, then it will be damaged. And now what we have to do, first of all we have to calibrate the pH meter before using, because without calibration maybe the result become altered. Now how we calibrate our pH meter? pH meter is calibrated by the buffer solution. There are two type of buffer present in our lab, that is 9 pH 7 and pH 4. Now what we have to do, first of all we have to set the temperature 25 with the help of temperature knob. Then what we have to do, we have to remove this knob, we have to remove this electrode from the KCl and wash with the distilled water. After that we have to wipe it with the filter paper. You have to take extra care during the handling of the electrode, because electrodes are very crucial. Now what we have to do, after wiping off the electrode with the filter paper, we have to dip this filter paper into a pH 7 and move the function knob. When the display shows 7 pH, we will stop it. After before moving the function key, we have to move these standby keys from the standby to the pH. Now after that what we have to do, we have to move the function knob and set the pH 7. Now after that, when these reaches to the 7, we have to again move the knob from pH to the standby node. Now we don't touch these two buttons, that is temperature knob and the function knob. Now what we have to do, we have to again wash these electrode with the distilled water and again wipe with the filter paper. Now we put these electrode into a pH 4 buffer, and again we move the knob standby from standby to the pH. And now what we have to do, we have to move the slope button and set the pH 7 as a pH 4 as a display show pH 4. We have to stop it, and now our pH meter is calibrated. Now if we want to take a pH of any sample, we have to take it same as it is. We have to wash with the electrode with the distilled water, and after that what we have to do, we have to wipe it and dip the electrode into a sample and take a pH. Now this is all about the pH meter. The next is your hot air oven. Hot air oven is used, the use of hot air oven is very different. Like it is used for the sterilization of glasswares, and it is also used for the removing of the moisture. Okay, so if we want to sterilize the glassware, we have to set the temperature at least 160 degrees Celsius for 30 minutes, and if we want to remove the moisture, then the temperature range will be 130 to 135 degrees Celsius, and the time is very first. The time range is about 2 hours. After 2 hours, we take a weight, and again we put the sample into a hot air oven at the same temperature and wait for 30 minutes, and again we take a weight, and this process is go on as a weight become constant, and every 30 minutes interval. This is the function of the hot air oven. Hot air oven also have a knob whose temperature range is 50 to 300 degrees Celsius. There is a main indicator, there is also a temperature indicator, and there is a display which show the temperature range. And also there in some hot air oven there is a thermometer placed which show the temperature range. Instead of these display, there is a thermometer knob present into a hot air oven, and it show the range of temperature. Now see what the hot air oven is. Hot air oven is double walled, insulated, keep the heat in and conserve the energy. The inner layer being poor conductor and outer layer being a metallic. This is all about the hot air oven. Now next is your vortex. Vortex is very simple instrument. It is simply used for the mixing of the small volume of liquid, and it consists of an electric motor and with drill shaft oriented vertically, and it is attached to a corked rubber piece in which we put our sample, and when the motor runs, the rubber piece accelerates rapidly in a circulation motion. See, this is your rubber piece. When the motor attached to the rubber piece is moved, these rubber piece is also accelerate, and when these accelerate, it will move the volume present in a test tube rapidly in a circular motion, and by this way the liquid is mixed. The next is your water bath. Water bath is simply used to maintain a very stable temperature, just like your incubator. Okay? Again, the use of water bath by two way. First is suppose you have to give heat to them. Suppose then we use water bath, and sometimes we have to use for a incubation. Okay. Water bath can hold often temperature within a tenth of a degree Celsius. The water is often circulated. Sometimes beads are used as a waterless option. Scientific water bath is also used for a commercial kitchen to cook. Okay. The function of this is all about the water bath. Again, the water bath have a temperature knob, the main switch, the temperature knob, main switch, indicator, temperature indicator, etc. Okay, this is the all function of the. And one more thing, there is a heating mantle present in a heating mantle and plate present in a hot air oven. So before filling the water, we have to take care about these two things. These heating mantles should be dipped into a water. Okay. The next, these plate will not float into a water. Float into a water. So these two things we have to take a care. Otherwise our water bath is not work properly. If the heating mantle is not dipped into a water, then it will be burned, and if the plate is floated into a water, then it will cause contamination. Now next is your rotary shaker. Rotary shaker is a very simple device. It is used to mix or agitate the small volume of solution, and this solution is used further into a centrifuge. The next is your laminar air cavity. The microbial experiment is done into the laminar cabinet airflow. Laminar consists of UV light and HEPA filter. HEPA filter does not allow the even the smallest microorganism to pass through into it. Okay, why this? Because the size of the HEPA filter is less than 0.1 micron and the smallest particle PPLO, its size is 3 micron. That's why the smallest microorganism will also not pass through the HEPA filter, and these UV light is used for the sterilization of the cabinet. Now what we have to do, we have to sterilize these cabinet with the 75% of ethanol. Okay, we clean these. So first of all, what we have to do, we have to clean these cabinet with the 70% of ethanol to remove the contamination. Now what we have to do, we have to switch on the UV light for 15 minutes. After 15 minutes, we switch off the UV light, and when we start our work, we switch on the airflow. And the function of the airflow is already told you, the role of the HEPA filter. This is all about the laminar air flow. The next is your autoclave. As we all know that autoclave is used for the sterilization of the media, glassware, etc. Glassware, medical instruments, etc. Now, what is the meaning of the sterilization? Sterilization is simply means that to remove or kill the all type of microorganism. As we all know that in autoclave what we do, we kill the all type of microorganism. There is a temperature, pressure and time combination. Temperature will be 121 degrees Celsius, pressure will be 15 psi and time will be 20 minutes. When this pressure reaches to the 15 psi, after that we count the 20 minutes. Okay, now the autoclave is discovered by the Chamberlain in 1879. Through a precursor known as a steam digester was created in 1679 by Dennis Pepsin. Now autoclave also have heating mantle and plate. So we have to take care that these heating mantles should be dipped into the water and plate should not float. The reason is same, not float. If the plate is float, it causes what? Contamination to the media, and if this heating mantle is not dipped, then it will be burned. This is all about the autoclave. Now colorimeter. Colorimeter is used for the measurement of the wavelength absorbed by or transmitted by the object. Colorimeter is basically used in various fields like chemistry, physics, biology, industry, and in practical term, colorimeter is typically consist of light source, series of filter. It is what? It is consist of light source, series of filter or sensor to isolate a specific wavelength, and a detector to measure the intensity of a light after it interact with the object. The device then provide color information in terms of like RGB, like red, green, blue or other color space. See, this is your colorimeter. Here the wavelength range are mentioned that if you consider that the wavelength of the red is 58, it is for example, it is the mention that then we set here a 58 and put our sample here in colorimeter tube, and here the display show the reading. This is all about the colorimeter. Now incubator. Incubator is, as we all know, that incubator is used for the microbial growth because it gives the proper environment for the growth of the microorganism, like temperature, humidity, concentration of carbon dioxide, okay, etc. Now, what is the role of the incubator? Incubator provide environment for the growth of the microorganism, okay, for the growth of the microorganism. As we all know that the microorganisms are grow at particular temperature, like bacteria, okay. So the bacteria should grow at 37 degrees Celsius especially. So we have to give the 37 degrees Celsius with the help of incubator. After that the bacteria should grow, okay. So this is all about the instrumentation, the instrument which we are using in our lab. If you have any query, let me know in the comment box, and you can also follow us like ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and you can also download our app from a Google Play. And this is a helpline number also mentioned, that if you have any query, you have to message your query in this helpline number. Thank you so much. I hope you understand today's topic.